Hello, my name is Morgan Shine, and I'll be discussing some of the work from my honors thesis, which is titled Biophysical Characterization and Computational Modeling of Histone mRNA Degradation Intermediates. Histone mRNA is unique because it's the only known metazoan mRNA that does not end in a poly-A tail. Instead, it ends in a highly conserved stem loop structure that's formed by Watson-Crick base pairing. The stem loop and the stem loop binding protein, or SLBP, control all aspects of histone mRNA metabolism. SLBP is phosphorylated for most of histone mRNA metabolism, except when the mRNA is degraded at the end of S phase. At the beginning of histone mRNA degradation, the stem loop is bound by SLBP and an enzyme called 3' human exonuclease, or 3' hexo, and this forms a ternary complex. 3' hexo removes 5 to 7 nucleotides from the 3' end of the RNA, and a cytoplasmic uridylyl transferase called TUT7 adds non-templated uridines, creating uridylated degradation intermediates, some of which are much more common in the cell than we would expect. And so the focus of my research is on improving our understanding of how these uridylated intermediates function in degradation by investigating how the trimming and uridylation of the stem loop affects its binding interactions with SLBP and 3' hexo. In this study, we have 3 major RNAs of interest. There's the wild-type stem loop, which represents the highly conserved 26 nucleotide sequence, the uridylated intermediate, or 5U, which has 7 nucleotides removed from the 3' end and 5 uridines added, and the truncated stem loop, or trunk, which has 7 nucleotides removed from the 3' end. In Figure 2 in the top middle section of the poster, we've shown with electrophoretic mobility shift assays that SLBP and 3' hexo are able to bind both the uridylated intermediate and the truncated stem loop despite 3' end trimming and uridylation. Once we experimentally validated that these complexes could form, we performed 18 different molecular dynamic simulations, each for 1 microsecond. Each of the 3 stem loops has been simulated in 6 different protein binding conditions, which include the RNA by itself in a water box, the RNA bound to SLBP in both of its phosphorylation states, the RNA bound to 3' hexo, and the RNA bound to both proteins in the ternary complex, again with both phosphorylation states of SLBP. Figure 3 in the middle of the poster summarizes some of the results from the phosphorylated ternary complex simulations. Here we're looking at root mean square deviation, or Rmsd analysis, which quantifies how different structures are from a common reference. In this case, the reference is the RNA from the phosphorylated ternary complex crystal structure. We've used this analysis to show that the wild-type stem loop and the uridylated intermediate adopt similar conformations to the initial reference structure, as indicated by their low RMSDs that overlap, but the truncated stem loop adopts a separate conformation that deviates much more from the reference, and this is indicated by its much higher RMSD, near 10 angstroms. The representative frames in Part D of Figure 3 further show that the uridylation in the 5U helps it maintain the overall shape it needs to participate in the ternary complex. In the final results section in the middle, at the bottom, we're looking at hydrogen bonds between G7 of the RNA and arginine 181 of SLBP. This interaction is significant because 2 hydrogen bonds between G7 and arginine 181 were the only base-specific interactions identified in the original ternary complex crystal structure. Mutagenesis studies have also shown that changing G7 to another nucleotide eliminates SLBP binding, and so this interaction is really critical for histone mRNA SLBP binding. We compared hydrogen bond occupancy values for each of the 3 stem loops in their phosphorylated and dephosphorylated ternary complex simulations, and we found that the wild-type stem loop and the truncated stem loop exhibit large decreases in occupancy for the 2 base-specific interactions when SLBP is dephosphorylated, but the uridylated intermediate exhibits large increases in occupancy. And so this suggests that uridylation of the stem loop actually helps maintain a key set of hydrogen bonding interactions with SLBP after SLBP has been dephosphorylated. Overall, our results support a model of histone mRNA degradation initiation in which uridylation of the stem loop helps maintain the overall structure and RNA-protein interactions required to maintain the ternary complex after SLBP dephosphorylation.